Hello, welcome to Zoominario. Today we have Ana Salgado. She was born in Ecuador and obtained a bachelor's degree in biological sciences at the Pontificia Catholic University of Quito in Ecuador. Then achieved a master's degree in behavior, evolution, and conservation at the University of Lausanne in Switzerland, and a PhD in wildlife research at the University of Helsinki in Finland. Now she's a postdoctoral researcher at LSU in the US, investigating the chemical changes on host plants and its implications on the plant-herbivore interaction. Thank you very much, Anna, for accepting the invitation.
Thank you for the invitation. Actually, I'm really excited to share with you all my research that I've been doing. Specifically, most of my research is about my PhD. Well, I will start. I will talk today about the ecological implications of host plant quality, in a specific for herbivorous insects. So just to start, well, in general, organisms have to develop in a very stressful environment, and because environments are very, very variable, they change on temperature and UV and pathogens, precipitation, and in different other aspects, and even crowding, for example. So the organisms that live in any ecosystem have to really respond to these conditions. And the way that normally we ecologists study this relationship between the environment and organisms is this direct interaction, where we see how one change in the environment will affect directly to another species. However, this is way much more complicated because the environment is not just one species, one ecosystem, no? It's a mix of it, and we have the ecosystem that affects first species, and this species is related to a second species. So these indirect effects are the main that I was very, very interested on my research. Apart of joining also the looking into the direct effects of the changes in the environment to the species. So I study specifically drought, and I define drought as the reduction in precipitation. And I mainly focus on this environmental stress because it's predicted to be more frequent and severe in certain areas around the globe. And what is expected is that the drought will change the food quality for certain organisms, in this case from herbivorous insects. And the insects will have to try to change their life history and their behavior in order to survive. So during my dissertation, I work in two parts. In the first part, I look into the effects of drought in the host plant, mainly looking into the nutritional part of the plant, how it's affected when the plants are under drought. In my second part, I try to relate that the effects of the host plant quality in the herbivore, looking into the preference and the performance of the insect. So for the first part of my talk here, I will start with the effects of drought on the host plant. So for this, I started with experiment where I set the plants from in a greenhouse, and I set them to put well water with normal amounts of water and a drought exposed amount of water. So it was a small reduction of water every 2 days, as you can see, and already on these two pictures, you can see that the plants develop completely different. And these already observations make me think that this could have effects on the butterfly. But first, so as I was interested to understand what's happening on the butterfly, I measured the nutrient content of these plants, especially because these are key components for the development and the reproduction of the butterfly. So one of the first results that I found from this experiment is that the carbon and nitrogen increases under drought conditions. So as you can see in these two graphs, the plants that are here on yellow have way much more carbon and nitrogen, suggesting that they are like the plants adapting differently to the drought stress. And this makes me think, well, then if eating from drought exposed plants will increase the herbivorous insect performance. And that's what I wanted to do in the second part of the project, where I basically look into the effects of the host plant quality on the herbivore. So for this, I took caterpillars and I fed them in two types of diet. So I fed them with well-water plants and with drought-exposed plants, and then let them grow. When I was looking then into the growth rate of these caterpillars, I found that eating in drought-exposed plants increased their growth rate, as you can see here in yellow. So it seems that the butterflies are doing much better when they are eating from these drought-exposed plants, because they have other components like carbon and nitrogen, specifically nitrogen, which is a very difficult component that's very limited in the nature. So it seems that drought will be very, very beneficial. But so far here, as you can see, I did experiments on conditions that were established in a greenhouse. So I was more interested on the ecological implications of feeding in these drought-exposed plants in the field. And then is when I worked for a really long time, during 4 years in my PhD, collecting data from this butterfly. So just a bit of a background of the butterfly. This is the Glamby fritillary butterfly, Melitaea quincea. It lives in Finland, but only in this tiny archipelago here in southern Finland. This is a classical metapopulation because it has several populations. It's different populations, so this is what's a metapopulation. And these populations are connected with migration, but they also suffer from extinction. That's why it's considered a classical metapopulation. And a bit about their way, how they grow in these conditions in Finland. So during the summer, the butterfly flies and female meets a male and then lay the eggs, and the caterpillars grow until the 5th instar, and they are very sessile. So basically, where the mother puts the eggs is where they develop. They move very, very few centimeters looking for host plants. Then when the winter arrives, the butterflies, they create this web and they live together, a group of 100 or 200 caterpillars. They create this web and they survive winter on this way. And then on the spring, the butterflies will do 2 more instars, and then they will continue with their life cycle. So what's important here is that you notice that the individuals that are in the very small individuals are very gregarious. And as I told you, like we can link where the females oviposit their eggs to the survival of the individuals, of their offsprings. So for my experiment, I took 12 localities in the islands and I surveyed them over 4 years. These 12 localities, and then I also used this long-term data that we have of the oviposition sites of the butterfly. So from 2009 until 2018, we collected all this information of where the caterpillars were found within each of these localities. And then this is easy to do it because in the field you will see, like you see in this photograph here, the caterpillars have this winter nest. This is what we call it, a nest. And when you're walking around the localities, you can find them very easily. So during autumn, there's a group of 50 field assistants that they go to the islands and they survey every single locality and they mark every single nest. Then after, in the spring, we have all these coordinates, so we are able to go back and look if the caterpillars are alive or not. So we register all this information and then we use this information to see what's going on with drought. Apart, during the last 4 years, I also collected information about the environment where these butterflies live. So I divided every single locality in these quadrats of 20 by 20 meters, and then I determined the host plant abundance and the drought sensitivity of the plants. I will mainly focus on these 2 components of the environment because they were the most important for understanding the preference of the butterfly. So one of the first results that I found, it was that females will prefer these areas within a locality where there is a great amount of host plants, as you can see here. So that's not very surprising because this butterfly, it only fits in 2 plant species in these islands. So basically they need from this host plant to survive. Then when we look into the drought sensitivity of the plants, we found that the females also have this preference to look and put their eggs in plants that suffer way much more drought than in areas where there is no, there is less drought. And this is also when we look to this time series that we have from 2015 to 2018, we also found that there are differences in a wet year or in a dry year, where all these preference for a drier areas is stronger in drier years. So this is very interesting for the biology of this butterfly. You have to remember that Finland is a country that's very, very up north in the latitude. So it's a cold country and they have a very, very short summer. So the butterfly species is very clever in the way that they know that they have to grow fast and continue with the cycle. So basically they prefer these drier and hotter areas where they can develop faster. So as you can see here, the females choose to oviposit in drier microhabitats with higher host plant abundance and especially in drier years. So another observation that we did from this experiment is that we found that there is a higher proportion of dry plants and the probability of survival over winter. So as drier is the area, the butterflies will do it much better for after the winter. And I say here that this happening in an average year. And I say that because as I was telling you, we have long-term data on the population of this butterfly. So from 1993 until 2018, the researchers have counted the number of nests and the number of individuals in the population. And as you can see here, for example, annually there is always variations and fluctuations on these populations with higher peaks, for example, here in 2018, but after there's a crash. But in 2018, it happened that there was a real crash on this population and happening just to find 100 number of nests within this island. I'm telling you, this island has 70 for 50 kilometers. So the number of individuals in the islands really dropped very, very drastically. And what we found is that this was linked to a very, very, very dry year. So in 2018, there was a heat wave passing over Europe, especially in the north part of Europe. So there was abnormal hot temperatures, and this causes that directly all the localities where the butterfly live were completely dying. So basically the butterfly kept doing the same, kept choosing these very dry, hot areas to try to develop, but the plants were not enough for feeding these caterpillars. So there it seems to be this mismatch when there is going to be changes with these abnormal temperatures in the future. And hopefully we are able to keep seeing some adaptations of this butterfly for these new differences, but that has to be done furthermore. So, but then when you see, I was looking on this part of the how host plant quality affects the individuals. I was just looking into simple compounds. No, I look into carbon, I look into nitrogen. However, when an insect is eating from their host plant, they encounter a zillion amount of compounds that are on the plants. And that's where I decided to join to this postdoc now in LSU in Louisiana, where I'm looking mainly into the metabolome of the plants to see how they respond to the environment. So now I'm working with this other plant. It's a grass, it's the common reed that you can find also in the Great Lakes. I'm looking mainly into the resistance and the tolerance of this plant. This plant is distributed all along the world. It's a cosmopolitan plant. However, there are different lineages that has been identified for this plant. And what is more surprising is that every single lineage has differences in the herbivore resistance. So here in North America, there is the European introduced lineages, but there is also the native lineages. And it seems that the European has been doing much better, especially in the Gulf. So this is our question: why is these plants are doing much better when they are attacked with this scale insect? So I'm looking mainly to the plant metabolome because that's the way that plants basically respond to the environment, to the stress. So with primary metabolites, the plants are able to look into to defend and respond to the abiotic stress. So for example, to heat, to drought, to salt, but they are also important for the plant development. In the other part, you have what's called the secondary metabolites that are mainly used for responding to other biotic factors like insect attacks or herbivores or other type of damages. So this is what I was interested to look and to try to explore a bit further. But there is also one more component that's important on plants, and it's mainly the mechanistical defense. And this is found on lipids, which are mainly on membranes. And they are also found to have important role on chemical defenses and communication. So basically for this postdoc, I'm trying to understand how these two will respond to differently on the lineages. So using machine learning techniques here, specifically I'm using random forest. I've been trying to identify from big data. I'm talking 2 data sets from 6,000 each data set of lipids and secondary metabolites to try to identify compounds that are important for the lineages. So I've been able to locate with random forest these 10 compounds that seems to be variable in between the lineages. And using these compounds, we've been able to distinguish the lineages. So we already see that there are differences on how every single lineage respond, how they interact and how they do it. Actually here, there is no differences on the environment because the plants were grown in the greenhouse. However, there seems to be genetical differences. What's interesting to see here is, if you notice, there is the North American native lineage, which is in red here, and the European invasive, which is already part here in the US from 100 and more years. That's the first record on the herbarium. So it seems that they share already compounds, and they are very, very similar on their compounds compared to, for example, the European lineage that's still in Europe. So continuing a bit more into this, this chemical part, we also wanted to know what was the richness, chemical richness of these plants. And we see that there are differences on mainly on evenness and not much on richness. As you can see in the first graph, you have richness, and most of the lineages seems to have the same amount of compounds, but there is one lineage that has lower richness. However, what we are really interested here is to see that this within a species, evenness it seems to be a very important chemical evenness seems to be very important for disentangling differences on the lineages. So this is just mainly the basic part. Let's say trying to understand how the chemistry changes over in a lineage, in a species, no? But there is still the other part that we are trying to do now is where we are trying to put the herbivores to feed from these plants and to see how the chemistry will change. And we also, in another project that I had last year, we manipulate also the microbiota of these lineages to see if there are differences on the changes of the chemistry of these plants and how this will be linked to the herbivores. But this is only, I cannot say much, just how I did the experiment because I'm still working with those results. And well, my take-home message is that, well, insects adaptation and survival depend tightly mainly on how plants respond to the environment. And this is basically because herbivorous insects are tightly to their host plant. So it's interesting that we start looking more into these indirect effects of the environment on how herbivorous insects will respond. But also to take into account that this is more complicated and we have also the direct effects. And with that, I want to thank everybody that worked with me during my career here in Ecuador, in Switzerland, Helsinki in Finland, and now to my supervisor, Dr. Cronin in LSU. And thank you, Claudia, for the invitation. Now a bit more into the nice part of talking about my country. So I come from Ecuador, and I'm from the highlands, and I'm right now here. And that's basically how I observe my city from the top hills. And it's a big city compared to where I live now in Louisiana. But well, just to let you know, Ecuador is a country that's in between Colombia and Peru, and we are surrounded with the Pacific. So we have 4 regions. The first one, the coast region. Then we have the highlands, where we have the Andes. And we are a country with volcanoes. We have a lot of volcanoes, and for us it's so normal when we have a volcano active. And then we have also part of the Amazon. So as you can see already, my country is very diverse on ecosystems. And this is one of the things that really attract me a lot, to try to understand how plants and animals interact. And one of the most iconic parts of Ecuador, and probably for biologists, is the Galapagos Islands. So it's a very touristic area, but for us, for biologists, it's very important because it helped to develop to Charles Darwin his theory of an evolution, and yeah, it's an iconic for us. And one fact that I'm not really proud, but it's always surprising for people in the US, is that I'm telling them, is like, yeah, our currency in Ecuador is the dollar. So since 2000, we have a really bad crisis, economic crisis, and is when we change our national sucre currency for dollars. So since then we have dollars, and so it's easy for people from the US to come here and visit us. So all this diversity of regions creates all this what's called the mega diverse country that we have. We are on the top list of mega diverse countries with so many plants and animals in general. Not to say with other understudy groups, no. Then one of the things that I think is very unique in my country, and especially now that it was New Year's, it's what's called the Noche Vieja, the New Year's Eve. So there's 2 events that I think that I have never seen this in another country. And the first one, as you can see in the first picture, we have this doll that represents the old year. So it can also represent, normally they put at faces of political characters, but it can also now be like comics or anything. And then at midnight, we go out after we do the huggings and the cheerings, and we go out and we burn these. So we represent that we burn all the bad things that happened last year, and we are looking for a new year. And then another thing that happens a lot, especially here in Quito, is that in Noche Vieja, in the New Year's Eve, guys they take costumes and they put themselves as widows. Now it's a bit different, like they use colorful clothes, but they tend to represent widows, and they go to the street and they beg for money in the cars. So that's a very unique thing that I have never seen, and it's very funny to go out. I actually like to go driving on the city and see all these people, special guys, like wearing these tight things and high heels. So it's very nice how they integrate these. And yeah, with that, I would like to thank you, Claudia, for the invitation again, and for everybody to be here. And I'm open for questions.
Thank you, Anna. I'm going to stop the recording.